A houseplant sits on a windowsill. Its leaves have begun to brown at the edges, curling slightly inward, the tips turning crisp and papery. The owner notices, assumes neglect, and waters it more frequently. The browning spreads. Frustrated, they move the plant to a different room, trim the damaged leaves, and wait. Within weeks, new leaves emerge with the same brown edges. Eventually, the plant is discarded. The assumption was always the same. Something went wrong, the plant was sick, and the damage could not be reversed. But the plant was never sick, it was speaking. Every discoloration, curl, spot, and wilt that appears on a leaf is not random damage. It is a physiological response, a visible translation of invisible stress. Plants cannot move when conditions become unfavorable. They cannot migrate toward water, flee from heat, or escape a pathogen. Instead, they communicate through their tissues. Brown tips signal water loss. Yellow leaves signal nutrient reallocation or root failure. Curling indicates either dehydration or chemical overload. Spots with yellow halos mark the boundary where a plant is defending itself against infection. For centuries, farmers and gardeners understood this instinctively. They could walk through a field and diagnose problems by observing leaf color, texture, and the pattern of damage. A farmer in 18th century Europe did not need a laboratory to know that yellowing lower leaves meant nitrogen was being pulled upward, or that curling edges meant the air was too dry. That knowledge was survival literacy. It allowed people to grow food, manage crops, and keep plants alive without access to chemical interventions or modern diagnostics. But over time, that diagnostic language faded from everyday practice. Modern plant care advice often treats symptoms as isolated emergencies rather than as part of a continuous biological conversation. A brown tip becomes a problem to fix with a product. A yellow leaf becomes a reason to change the soil or buy fertilizer. The underlying logic, the reason the symptom appeared in the first place, is rarely explained. Yet the language still exists. Every symptom corresponds to a specific internal process, and those processes follow consistent physiological rules. Understanding them transforms plant care from reactive guesswork into informed diagnosis. The most commonly misunderstood symptom is also the most common. Brown tips appear on houseplants in nearly every home, across every climate, in every season. Most people assume disease or poor genetics. The actual cause is far simpler. The plant is losing water faster than its roots can replace it. The scientific term for this condition is transpirational stress, and it begins at the leaf edges because those cells are the furthest from the plant's vascular system. Water moves from the roots upward through narrow tubes called xylem vessels, traveling through the stem and into the leaf veins before finally reaching the cells at the leaf margins. When a plant loses water through its leaves faster than the roots can absorb it from the soil, the cells at the end of that transport line are the first to run dry. Once a cell dies from dehydration, it does not recover. The tissue turns brown and becomes papery because the cell walls collapse after the water inside them is gone. Cutting off the brown tip does nothing to solve the problem because the underlying imbalance between water loss and water absorption remains unchanged. The plant will simply produce new brown tips on the next set of leaves until the environmental cause is addressed. The most common triggers are low humidity, inconsistent watering, and exposure to heating vents or fans that blow dry air directly onto the foliage. In each case, the rate of evaporation from the leaf surface exceeds the rate at which roots can pull moisture from the soil and transport it upward. The solution is not cosmetic trimming or fertilizer. It is environmental adjustment, raising humidity levels, watering more consistently before the soil dries completely, or moving the plant away from direct airflow. Brown tips are not a disease, and they are not caused by neglect in the way most people assume. They are a mechanical problem of water transport, visible at the point where the system fails first. Another commonly misread symptom appears lower on the plant, where older leaves begin to yellow while the upper foliage remains green. Most gardeners see this and assume something is wrong: nutrient deficiency, root damage, or the beginning of systemic decline. In many cases, the plant is doing something entirely deliberate. It is reallocating nitrogen from its oldest, least efficient leaves upward to support new growth at the top. The process is called senescence, and it is a form of programmed cell death that allows the plant to recycle nutrients internally rather than losing them when a leaf falls. Nitrogen is one of the most metabolically expensive elements a plant uses. It is required for chlorophyll production, enzyme function, and the construction of proteins that drive photosynthesis. When nitrogen becomes limited, either because the soil contains less of it or because the plant is growing rapidly and using it faster than it can be absorbed, the plant pulls nitrogen out of older leaves and redirects it to younger, more productive tissue. The older leaves yellow because chlorophyll breaks down once nitrogen is withdrawn, and without chlorophyll, the green pigment disappears. If the yellowing is gradual, affects only one or two lower leaves at a time, and the rest of the plant appears healthy, there is nothing to fix. This is normal metabolic behavior, not pathology. However, if yellowing spreads rapidly upward and affects multiple leaves simultaneously, that signals a different problem entirely, one that involves the roots. When yellowing appears across the entire plant at once, rather than selectively on lower leaves, the roots are almost certainly involved. Overwatering is the most frequent cause, but the mechanism is counterintuitive because the plant is not drowning in water. It is suffocating from a lack of oxygen. Roots require oxygen to function, and they absorb it from the air pockets that exist naturally between soil particles. When soil stays wet for too long, those air pockets collapse and fill with water, creating an environment where root cells cannot access the oxygen they need to sustain cellular respiration. Without oxygen, root cells begin to die, and as the root system deteriorates, the plant loses its ability to transport water and nutrients upward into the leaves. Chlorophyll production shuts down because the plant can no longer access the nitrogen and other elements required to maintain it. The leaves yellow uniformly across the entire plant because the failure is systemic rather than localized. This is why adding more water to a yellowing plant often accelerates its decline rather than reversing it. The problem was never a lack of water, it was a lack of oxygen in the root zone. Once root tissue begins to rot, anaerobic bacteria and water molds, particularly species from the genera Phytophthora and Pythium, colonize the damaged tissue and spread through the root system. The roots turn brown or black, become soft and mushy, and may emit a foul smell as decomposition progresses. At that stage, the only intervention that can save the plant is removing it from the soil, cutting away all damaged roots, treating the remaining healthy tissue with a diluted hydrogen peroxide solution to kill residual pathogens, and repotting in fresh, well-draining substrate. If the rot has progressed too far and no healthy roots remain, the plant cannot be saved. The diagnostic test is simple. Unpot the plant and inspect the roots directly. Healthy roots are white or light tan and firm to the touch. Rotting roots are dark, soft, and structurally compromised. Another symptom that is frequently misinterpreted is leaf curling, but the direction of the curl determines the cause. When leaves curl inward, rolling their edges toward the upper surface of the leaf, the plant is reducing its exposed surface area in response to water loss. This is not damage, it is a controlled physiological defense mechanism. By curling inward, the plant decreases the total area through which moisture can evaporate, slowing the rate of water loss until conditions improve. This response is most commonly triggered by underwatering, very high temperatures, or extremely low humidity. Some tropical species can appear severely wilted and curled within a single afternoon of dry conditions, then fully recover within hours once water is provided. The key diagnostic test is whether the curl reverses after watering. If it does, the problem was water stress and the plant has responded appropriately. If the curl does not reverse, root damage may already be preventing water uptake even when moisture is available in the soil. Downward curling, where leaf edges roll under toward the lower surface of the leaf, is a different symptom with a different cause. It is most frequently associated with overfertilization, specifically excess nitrogen. When a plant receives more nitrogen than it can metabolize, it pushes out rapid, soft new growth that the leaf structure cannot fully support. The edges curl downward under their own weight, because the tissue is too weak to remain flat. Heat stress can produce the same response in some species, but the distinction matters because the corrective action is opposite. If curl is caused by underwatering, the solution is to add water. If it is caused by over-fertilization, the solution is to stop adding nutrients entirely and flush the soil with plain water to dilute accumulated salts. Brown or black spots surrounded by a ring of yellow tissue are the signature of fungal infection, and the yellow halo is not decorative. It marks the boundary between dying infected tissue and the living cells the plant is attempting to defend. Fungal pathogens spread through water, specifically through water that sits on leaf surfaces long enough for spores to germinate and penetrate the leaf tissue. Splashing during watering, high humidity combined with poor air circulation, or leaves touching each other in wet conditions all create ideal environments for fungal spores to establish infection. Once a fungus colonizes a leaf, it spreads outward from the initial infection site, killing cells as it moves and triggering a defensive response in the surrounding tissue. The yellow halo represents the zone where the plant is mobilizing chemical defenses to slow the spread, but infected leaves do not heal. The spots expand until the leaf dies and falls from the plant. The most important intervention is not a fungicide. It is changing the watering method and improving air circulation. Water should be applied at the base of the plant rather than overhead, and affected leaves should be removed immediately to reduce the number of spores available to spread to healthy tissue. Fungicides can slow the progression of infection, but they cannot reverse damage that has already occurred. A very different pattern of damage appears when tiny pale dots begin to speckle the upper surface of leaves in a stippled pattern. Each dot marks a single cell that has been punctured and drained of its contents by spider mites, which are not insects but arachnids closely related to spiders and ticks. This distinction matters because standard insecticides designed for insects are usually ineffective against mites. Spider mites are extraordinarily small, often less than half a millimeter in length, and they feed by piercing individual leaf cells with needle-like mouthparts and extracting the fluid inside. A single female can lay up to 100 eggs in her lifetime, and in warm conditions those eggs hatch within 3 days, allowing populations to explode rapidly. The undersides of affected leaves are where mites live, feed, and reproduce, and fine webbing becomes visible once populations grow large enough. The fastest diagnostic test is to run a piece of white paper beneath a suspected leaf and tap the leaf sharply. Mites will fall onto the paper and move, appearing as tiny specks that crawl across the surface. A different metallic appearance signals a different pest. When leaves develop a silvery, bronze, or pale gray sheen across their upper surface, the cause is almost always thrips. These insects are slightly larger than spider mites but still very small, most measuring less than 2 millimeters in length. They feed by rasping through the outer layer of the leaf to access the cells beneath, and the silvery appearance comes from the empty air-filled cells left behind after their contents are consumed. Thrips are fast-moving and difficult to see without magnification, but they can be detected by shaking a branch over a white surface and watching for small elongated insects that move quickly. Unlike most houseplant pests, thrips are capable of flight, which allows them to spread rapidly from one plant to another and infest an entire collection within weeks. They also transmit plant viruses as they feed, which makes early detection particularly important. Both spider mites and thrips thrive in dry indoor environments, and infestations often begin during winter when heating systems lower indoor humidity and weaken plant defenses. A white or gray powder that can be rubbed off the surface of a leaf is powdery mildew, a fungus that behaves differently from most other plant pathogens because it does not require wet leaves to grow. Instead, it thrives in conditions where the air itself is humid but the leaf surfaces remain dry, which is why it appears most often in late summer or in indoor environments where temperature fluctuates and air circulation is poor. The white coating is not a symptom of infection. It is the fungal body itself, growing on the surface of the leaf and drawing nutrients from the cells beneath. As the fungus spreads, it weakens the leaf tissue, and severely infected leaves eventually yellow and drop from the plant. Diluted baking soda solution, neem oil, or potassium bicarbonate sprays can disrupt the surface pH that the fungus requires to survive, and removing badly affected leaves slows the spread to healthy tissue. One of the most persistent myths in houseplant care is that misting leaves increases humidity and benefits the plant. But misting does not raise ambient humidity in any measurable way. It only deposits water droplets on leaf surfaces, which creates ideal conditions for fungal and bacterial growth. The air circulation and rapid evaporation that prevent disease in outdoor environments do not exist in still indoor spaces, and water sitting on leaves becomes a vector for infection rather than a benefit. A very different kind of damage occurs when leaves lose their deep green color and become uniformly pale, bleached, or faded without any specific spots or patterns. This is called photobleaching, and it happens when intense light destroys chlorophyll molecules faster than the plant can produce them. Chlorophyll absorbs light energy during photosynthesis, but when light intensity exceeds the plant's capacity to process it, the chlorophyll molecules themselves break down under the stress. This occurs most often when a plant accustomed to indirect light is suddenly moved to a south-facing window or placed outdoors in full sun without gradual acclimatization. The damage is often irreversible in the affected leaves, though moving the plant to a location with filtered or indirect light stops further bleaching. Some species, particularly those native to tropical understory environments, can never adapt to full sun, regardless of how gradually they are introduced to it because their physiology evolved in low-light conditions. The opposite problem produces a different visual signal. Leaves that are darker than normal, deep, almost black-green, are not a sign of exceptional health, but rather an indication that the plant is compensating for insufficient light by producing excess chlorophyll to capture whatever light is available. This is the same mechanism that gives deep-sea algae their intense pigmentation, and while the plant remains functional in this state, it typically grows slowly, produces smaller leaves, and builds softer, weaker tissue that is more susceptible to fungal infection and pest damage. Moving the plant to a brighter position gradually returns leaf color to normal, and new growth will appear noticeably lighter and larger as the plant adjusts to higher light availability. A plant that wilts even though the soil remains consistently wet is experiencing root failure, and this symptom confirms that the root system has already been compromised beyond the point where it can transport water upward. The most common cause is root rot, a condition where roots have been kept in waterlogged soil long enough for anaerobic bacteria and water molds to colonize and destroy the root tissue. Without functional roots, the plant cannot move water from the soil into the stems and leaves regardless of how much moisture is available. Adding more water at this stage accelerates the destruction rather than reversing it because the problem is not water availability, but root function. The only intervention that can potentially save the plant is unpotting it, cutting away all dark, mushy, or foul-smelling roots, treating the remaining healthy tissue with diluted hydrogen peroxide to kill residual pathogens, and repotting in fresh, dry, well-draining substrate. If no healthy roots remain, the plant cannot recover. A sticky coating on leaf surfaces that feels like honey or syrup is not produced by the plant itself. It is honeydew, a sugar-rich waste product excreted by sap-sucking insects including aphids, scale insects, mealybugs, and whiteflies. These insects feed by inserting their mouthparts through the leaf surface into the phloem, the plant's internal transport system, and extracting plant sugars continuously. The honeydew they excrete coats the leaves below and attracts a secondary problem called sooty mold, a black fungus that grows on the sugar residue and blocks light from reaching the leaf surface. The mold itself does not infect the plant, but it reduces photosynthesis by covering the leaf and preventing light absorption. Treating the insect infestation removes the source of honeydew, and the sooty mold clears naturally once no new sugar is being deposited. Soft, translucent patches that feel waterlogged to the touch are caused by cell death from cold damage, and unlike most plant problems that develop gradually over days or weeks, cold injury can happen within minutes. It occurs when a tropical houseplant touches a cold window pane in winter, when an outdoor plant is caught by sudden frost, or when chilled water is poured directly onto warm leaves. The cells rupture because the water inside them expands as it freezes or chills rapidly, and the translucency comes from the collapse of the cell walls after the internal structure is destroyed. There is no treatment for cold-damaged tissue. The affected areas eventually turn brown or black and die. The same symptom can also appear from bacterial soft rot, but bacterial infections typically spread more rapidly and produce an unpleasant smell as the tissue decomposes. For most of human agricultural history, the ability to read plant symptoms was not specialized knowledge. It was survival literacy. A farmer walking through a field in the 18th century did not need a laboratory to diagnose nitrogen deficiency, water stress, or fungal infection. The information was visible in the leaves themselves, encoded in color, texture, curl, and the pattern of damage. That diagnostic language allowed people to grow food, manage crops, and keep plants alive without access to chemical interventions or modern testing equipment. It was practical knowledge passed down through generations, refined through observation, and essential to anyone who depended on plants for survival. Over time, as agriculture industrialized and plant care became increasingly product-driven, that language faded from everyday practice. Symptoms began to be treated as isolated emergencies rather than as part of a continuous biological conversation between the plant and its environment. A brown tip became a reason to buy fertilizer. A yellow leaf became a signal to change the soil. The underlying logic, the specific physiological process that produced the symptom, was rarely explained. But the language still exists and it follows consistent rules. Brown tips signal transpirational stress. Water leaving faster than it arrives. Yellow lower leaves signal senescence. Nitrogen being reallocated upward. Systemic yellowing signals root failure. Oxygen deprivation in waterlogged soil. Inward curl signals water conservation. Downward curl signals over-fertilization. Spots with yellow halos signal fungal defense. Stippling signals spider mites. Silvery sheen signals thrips. Each symptom corresponds to a specific internal process, and understanding those processes transforms plant care from reactive guesswork into informed diagnosis. Plants do not fail randomly. They respond predictably to environmental pressure, and those responses are visible to anyone who learns to read them. The knowledge was never lost, it was only forgotten, and what was forgotten can be remembered.